Welcome to the Axon Data Suite. I'm Ryan Carelli, a data scientist at Deepcell. In this video, we'll analyze a Deepcell dataset of the morphological changes associated with cell death. Jurkat T lymphoblasts were treated to induce apoptosis and necrosis. After treating cells, we measured the proportions of cells undergoing apoptosis or necrosis by flow cytometry, using an Axon-5 as a marker of apoptosis and cytox co-expression as a marker of necrosis to establish ground truth. When we color embeddings by ground truth label, we can see the distribution of cells from each of 5 treated conditions: untreated, camptothecin treated, staurosporin treated for 2 different intervals, and heat treated. We've included the percentage of cells from each condition in each cell death state as determined by flow cytometry. By toggling display images in the bottom, we can visualize the morphological changes identified by Deepcell's embedding model. Each treated condition has a distinctive morphological signature associated with treatment. Untreated cells, in purple, are concentrated in the top left corner of the plot, and these cells look viable with minimal granularity. Camptothecin treatment, in red, induces apoptosis. At 6 hours of 10 micromolar treatment, 30% of cells are in mid-phase apoptosis. Cell morphology has shifted, including swelling and increased granularity. Staurosporin treatment induces necrosis. At 3 hours of 2 micromolar treatment, shown in orange, 14.5% of cells remain viable, which can be seen by the overlap of cells with the untreated condition. The remainder of cells are in apoptosis or necrosis, with 14% in late necrosis. This is reflected in the shift from the healthy morphology to morphology showing the beginnings of nuclear condensation. By 18 hours of staurosporin treatment, shown in green, very few viable cells remain. Although the proportion of cells in apoptosis has only increased slightly, according to the flow cytometry measurement, the morphological difference is striking, and these cells are clearly at the end of their lives. After 5 minutes of 70 Celsius heat treatment, shown in blue, almost all cells are in early necrosis. The morphology changes from heat treatment are distinct, with the greatest degree of swelling and of granularity. Now I'll select a group of necrotic cells from the heat-treated condition. I'll pin these cells and call them necrosis. For comparison, I'll select a group of cells from the viable group, which I'll pin and call viable. Cell groups can be compared quantitatively to identify the distinguishing morphological features using the Compare tab. So I'll select the viable and necrosis groups. And by looking at a morphometric feature called small set of connected dark pixels integral, we can quantify the difference in granularity between these cell groups. The apparent difference in granularity in the necrosis condition is seen clearly in the distribution plot. An exciting aspect of the REMI platform is that now that the cell groups have been defined, we can sort viable cells from each group for characterization using functional assays. This gives us the ability to study the connection between phenotype, molecular, and morphological cell states. We've demonstrated the REMI platform's ability to characterize cell death pathways. Jurkat cells treated to induce apoptosis and necrosis are clearly distinguishable by morphology. These different morphologies can be selected and isolated for functional characterization. If you're interested in exploring the data yourself, click the Explore Data button to begin.